Topic of Pharmaceutical and Forensic Chemistry. Trace evidence analysis. Trace evidence. Trace evidence is created when objects make contact. The material is often transferred by heat or induced by contact friction. Since then, forensic scientists use trace evidence to reconstruct crimes and to describe the people, places, and things involved in them. The importance of trace evidence in criminal investigations was shown by Dr. Edmund Locard in the early 20th century. Trace evidence is based upon the principle known as Locard's exchange principle. Sir Edmund Locard (1877–1966) gave two principles. 1. Every contact leaves a trace. 2. Exchange of trace is usually a two-way process. Classification. Trace evidence may be grouped into 1. Biological evidence. 2. Non-biological evidence. Biological evidence. These originates from human, animal or plant source and include mainly 1. Body fluids. 2. Body tissues. 3. Stains. Body fluids includes blood, semen, saliva, urine, vomit, cerebrospinal fluid, etc. Such fluids may be found in the form of stains at the scene, clothes, and body of victim or assailant. Other biological materials. Human hair from head and body. Scrapings from the undersurface of nails which contains epithelium of skin of assailant or victim deposited during assault. Non-biological evidence. These comprise of 1. Items of personal use. 2. Other materials found in the environment. These personal items are clothes, spectacles, purse, wristwatches, or other things in use. The environmental materials are fibers from carpet, vegetation in the garden, grit particles from the road. Other non-biological materials consists of bottles, tablets, bullets or pellets, weapons of offense or any other thing used in the crime. Collection of specimens. The person must have the knowledge as to a. What is to be collected, b. From where to be collected, c. How much to be collected, d. How it is to be collected. A proper material from a proper site in a proper quantity by a proper method that is proper collection. Preservation. The preservation of specimens is done normally by two processes: desiccation or dehydration. Freezing. A proper preservative, in proper quantity having proper concentration, that is proper preservation. Preservation in different situations. In histopathological examinations we need a preservative which should preserve and prevent autolysis and harden the tissues. For chemical examinations we only require preservation. For serological examinations we also need anticoagulative activity. A good preservative. It should be also chemically inert. It should be easily available. It should be cheap. It should be effective for the purpose. It should not react with the container and specimen. Normally used preservatives. For chemical analysis, supersaturated saline is used. It acts by dehydration, that is, it extracts water from the tissues and they get preserved. For histopathological examinations 10% formal saline, that is 10% formalin in saturated saline, or 95% alcohol is used. For serological examinations anticoagulants used are calcium, oxalate, heparin, etc. Type of analysis: chemical, histopathology, serology, material, plastic, glass, sterilized glass test tube, size and shape, large mouth jars, according to size of organ, test tube, lids, plastic lids, glass lids, glass or rubber stopper, preservative, saturated saline, 10% formal saline, anticoagulant, labeling. Each container should be labeled indicating number of jar, 1, 2, 3. Should be mentioned. Name of specimen, site of collection. Preservative used. PMR/MLC no. Date and time. Name of person. Examination required. Name of doctor, signature and official seal should be affixed. Sealing of specimens. All the specimens should be sealed with sealing wax to avoid tempering and to maintain secret chain of custody. All the containers or jars should be packed in one box, and the box should be labeled indicating address to be sent. Arrow showing upside box should be sealed. Box should accompany a forwarding letter. Dispatch of the specimens. The only condition in which the specimens are allowed to be kept is that when the autopsy is conducted at odd hours, when the lab is closed. Specimens should be collected, preserved, and dispatched immediately after autopsy. Safe storage. The specimens should be kept in lock and key in the freezers. The seal should be intact, it should be compared with the specimen of the seal provided. The specimen should be registered. Transportation. The specimens should be transported through special messenger by hand, through railway parcel, through postal parcel, through police. Chain of custody. Any and all who handle the specimens at a particular instant should be able to certify the extent of handling, that is, what was collected, where it was collected, to whom it was handed over, when it was handed over, for how long it remained in custody. Chain of custody should be maintained to avoid adulteration and tempering. Chain of custody should be maintained till the specimens are presented in the court. Handling should be certified, that is. Principles of chain of custody. Shorter the chain, better it is by involving less and less persons. Every surrender should be on a written receipt. Surer the chain, better it is. Thank you.